Hello friends. Welcome to your blessed YouTube channel. Today we are going to learn about immunity. First let us know definition of immunity. Every day we are exposed to large number of infectious agents. However, only a few of these exposures result in disease. Why? This is due to the fact that the body is able to defend itself from most of these foreign agents. Thus overall ability of the host to fight the disease-causing organisms, conferred by the immune system, is called immunity. Immunity is of two types, 1. Innate immunity and 2. Acquired immunity. Innate immunity is non-specific type of defense that is present at the time of birth. This is accomplished by providing different types of barriers to the entry of the foreign agents into our body. Innate immunity consists of 4 types of barriers. These are 1. Physical barriers. Skin on our body is the main barrier which prevents entry of the microorganisms. Mucous coating of the epithelium lining the respiratory, gastrointestinal and urogenital tracts also help in trapping microbes entering our body. 2. Physiological barriers. Acid in the stomach, saliva in the mouth, tears from eyes all prevent microbial growth. 3. Cellular barriers. Certain types of leukocytes, WBC, of our body like polymorphonuclear leukocytes, PMNL neutrophils and monocytes and natural killer type of lymphocytes in the blood as well as macrophages in tissues can phagocytose and destroy microbes. 4. Cytokine barriers. Virus-infected cells secrete proteins called interferons which protect non-infected cells from further viral infection. Acquired immunity, on the other hand, is pathogen-specific. It is characterized by memory. This means when our body encounters a pathogen for the first time, it produces a response called primary response which is of low intensity. Subsequent encounter with the same pathogen elicits a highly intensified secondary or anamnestic response. This is ascribed to the fact that our body appears to have memory of the first encounter. The primary and secondary immune responses are carried out with the help of 2 special types of lymphocytes present in our blood, i.e., B lymphocytes and T lymphocytes. The B lymphocytes produce an army of proteins in response to pathogens into our blood to fight with them. These proteins are called antibodies. The T cells themselves do not secrete antibodies but help B cells to produce them. Each antibody molecule has 4 peptide chains. 2 small called light chains and 2 longer called heavy chains. Hence, an antibody is represented as H2L2. Different types of antibodies are produced in our body. Agar, IgM, IgA, IgA, some of them. A cartoon of an antibody is given in figure. Because these antibodies are found response. This is one of the 2 types of our acquired immune response, antibody-mediated. The second type is called cell-mediated immune response or cell-mediated immunity, CMI. The T lymphocytes mediate CMI. Very often, when some human organs like heart, eye, liver, kidney fail to function satisfactorily, transplantation is the only remedy to enable the patient to live a normal life. Then a search begins to find a suitable donor. Why is it that the organs cannot be taken from just anybody? What is it that the doctors check? Grafts from just any source, an animal, another primate, or any human beings cannot be made since the grafts would be rejected sooner or later. Tissue matching, blood group matching are essential before undertaking any graft, transplant, and even after this the patient has to take immunosuppressants all her life. The body is able to differentiate self and non-self, and the cell-mediated immune response is responsible for the graft rejection. Active and passive immunity. When a host is exposed to antigens, which may be in the form of living or dead microbes or other proteins, antibodies are produced in the host body. This type of immunity is called active immunity. Active immunity is slow and takes time to give its full effective response. Injecting the microbes deliberately during immunization or infectious organisms gaining access into body during natural infection induce active immunity. When ready-made antibodies are directly given to protect the body against foreign agents, it is called passive immunity. Do you know why mother's milk is considered very essential for the newborn infant? The yellowish fluid colostrum secreted by mother during the initial days of lactation has abundant antibodies agar to protect the infant. The fetus also receives some antibodies from their mother through the placenta during pregnancy. These are some examples of passive immunity. Vaccination and immunization. The principle of immunization or vaccination is based on the property of memory of the immune system. In vaccination, a preparation of antigenic proteins of pathogen or inactivated, weakened pathogen, vaccine are introduced into the body. The antibodies produced in the body against these antigens would neutralize the pathogenic agents during actual infection. The vaccines also generate memory, BNT cells that recognize the pathogen quickly on subsequent exposure and overwhelm the invaders with a massive production of antibodies. If a person is infected with some deadly microbes to which quick immune response is required as in tetanus, we need to directly inject the preformed antibodies. Or oxane, a preparation containing antibodies to the toxin. Even in cases of snake bites, IH inject on which is given to the patients, contain preformed antibodies against the snake venom. This type of immunization is called passive immunization. Recombinant DNA technology has allowed the production of antigenic polypeptides of pathogen in bacteria or yeast. Vaccines produced using this approach allow large-scale production and hence greater availability for immune cellin, e.g., hepatitis B vaccine produced from yeast.